Assalamualaikum and salam sejahtera to all examiner, to all lecturers and my fellow friends. My name is Muhammad Jais bin Sarifuddin with metric number 72363. Today I will present my final year project proposal. The title of my final year project is In Silico Characterization and Primer Design of Late Embryogenesis Abundant LEA Gene in Kelampayan. This slide was prepared by me, Muhammad Jais bin Sarifuddin, and supervised by Associate Prof. Dr. Ho Wei Singh. A little bit introduction about my proposal. Neolamarkia cadamba, also known as Kelampayan, is one of the species that belongs to Rubiaceae family and is in order of Genthernellus. Kelampayan is a tropical tree that is native to Southeast and South Asia. In Malaysia, Kelampayan is one of the important trees on the plantation. It has been chosen as one of the fast-growing plantation species for Sarawak planted forest development. There are a lot of uses of Kelampayan, such as plywood, pulp and paper furniture, raw material for perfume and alkaloid. Late embryogenesis LEA protein are extremely hydrophilic, glycine-rich protein with antioxidant, metal ion binding, membrane and protein stabilization, hydration, buffering, and DNA and mRNA interaction properties. They are necessary for the growth and development of plants as well as protecting cells from abiotic stress caused by environmental stress, notably drought. This discovery, as well as assumption that creating the tolerant cultivator would be immensely useful in agriculture, have full and search for anti-drought inducible genes and their characterization. The LEA protein was identified during the late embryogenesis and during the plant's development period. It was discovered in the roots, stems, and other organs. Molecular genetics has become one of the biggest research on plant research. Understanding of the genomic of the plants can improve the plant species. In silico and the primer design is one of the molecular genetic research. In silico refers to the experiment that is performed using a computer or via a computer simulation. Using the in silico method, a portion of the data sample with specified genomic, biological, and or clinical significance were imported and integrated to create gene expression profile and connect them to DNA methylation and protein expression. How about primer? Primer is a short synthetic oligonucleotide that is employed in a variety of molecular methods, including PCR and DNA sequencing. To design the primer of the LEA gene, the online primer designing software is available to pick the primer from the sequence of the gene. The objective of this study is to in silico characterization the LEA gene in the Kelampayan tree for a better understanding of the gene in Kelampayan. Another objective of this study is to design the primer for the LEA gene to be used in the further study of LEA gene in Kelampayan. We move on to the literature review for this proposal. The first thing is the species study. The species study is a Neolamarkia cadamba. Boza is also known as Kelampayan tree, and it is a local name of that species. There is also another name in the common English names such as barflower tree, lichen, pine, and laran are a part of Rubiaceae family which are angiosperm. Since 1930, the species botanical name has been subject of the lengthy taxonomic debate. Kelampayan is a larger, fast-growing deciduous tree with quality that provide an yearly economic return in 8 to 10 years. As we can see here, this is the picture of the Kelampayan tree, and in my right hand, this is the bark of the Kelampayan tree. This is the picture of the Kelampayan flowers and the Kelampayan tree. Let embryogenesis abundant LEA protein. LEA protein that are found in various tissues of abiotically stressed plant, some bacteria and invertebrates. LEA protein were first characterized as accumulating light in development of plant seeds. LEA protein are glycine-rich, extremely hydrophilic protein with antioxidant, metal ion binding, membranes and protein stabilization, hydration, buffering, and DNA and mRNA interaction capability. This class of protein is recognized for having intrinsically disordered structure and is mostly expressed when there is a lack of water. These proteins are linked to water shortfall resulting from alteration in the environment as well as water restriction applied during plant development under ideal growth conditions such as during the generation of seed and pollen grain or at key phase of growth of the shoots and roots. LEA proteins are known for having a skewed composition of amino acid as a result to get hydrophilicity and thermal solution stability. The amino acid sequence of the LEA protein was analyzed and it was discovered that there are 7 distinct groups of the LEA protein. Its set determine the various characteristics of LEA protein. Membrane stability is another role proposed for LEA protein. Ion sequestration has been hypothesized as an additional activity for this protein, especially for the LEA2 and LEA4 protein, where histidine containing motif appear to be critical for divalent cation binding. Next is in silico characterization. In silico process is performed using a computer or through a computer simulation. The phrase in silico is pseudo-Latin, which is referred to as silicon in computer chips. The term in silico is a recent term that refers to computer-assisted testing and is connected to the more well-known biology, in vivo and in vitro terminology. In silico technique including database, quantitative structure activity correlation, pharmacopers, homology models, and other molecular modeling approaches, as well as machine learning, data mining network analysis tool, and computer-based data analysis tool. Next is primer. A primer is a short synthetic oligonucleotide that utilized in molecular method like PCR and DNA sequencing. This primer is made from the reverse complement of a segment of template or target DNA that we want the primer to enable. Tag polymerase, like other DNA polymerase, can only synthesize DNA if it is provided with a primer, which is a short nucleotide sequence that acts as a template for DNA synthesis. The experimenter determined the DNA area that will be duplicated or amplified by the primer chosen in the PCR procedure. PCR primer are single-stranded DNA fragment with a length around 20 nucleotides. Next, we will move on to the materials and method of this research. For the in silico characterization, data mining is one of the first steps. The sequence will be obtained from the transcriptome database. The region of clampion will be predicted using a contig mapping approach based on express sequence tag EST. Identification of LEA clampion also will be done. To identify the LEA in clampion, the nucleotide sequence that given will be used as a query sequence. BLAST will be carried out using the NCBI gene bank database. After we conduct the data mining and identification of the LEA gene in clampion, the phylogenetic analysis of LEA evolution will be done. By using the NCBI database, the LEA gene sequence or amino acid sequence of the different plants will be collected. Then, from the gene sequence and amino acid sequence that will be obtained, the phylogenetic tree will be constructed using the MegaX software to contrast the evolutionary relatedness of the taxa. As we can see here, this is the preview of the MegaX software. The gene sequence will be uploaded in the FASTA form to create the phylogenetic tree. For the next, domain and motif analysis. The concept domain database in NCBI will be applied to analyze the presence of the LEA gene in clampion. It will be used to determine the domain analysis of the gene in clampion. As we can see at the picture, it is the preview of the NCBI concept domain database. For the motif analysis of the LEA gene in clampion, we will use the MIM for the motif analysis. We move on to the primer design. Primer of the LEA gene of clampion will be obtained using the Primer 3 Plus software. As we can see right here, this is the Primer 3 Plus software overview. The gene sequence will be uploaded in the FASTA form, and the annealing temperature, melting temperature, and the GC content of the primer will be set in the primer 3 plus software. After the primer was obtained, the data analysis of the primer will be done. The characteristic of the primer will be analyzed using the online software, which is Oligo Analyzer tool. As we can see at here, the hairpin, the structure of the primer can be investigated using this software. Next, we will move on to the expected outcome of the study. The study is expected to get success in silico characterization of LEA gene in Kelampaian by using the online bioinformatics software. The study is expected to discover the phylogenetic tree of the LEA gene in Kelampaian with other species, and the domain and motif analysis of the LEA gene in Kelampaian. Besides that, the study is expected to get successful and available primer that can be proper used in the molecular experiment purpose, especially for the PCR. This is the work schedule of the research activity. Cukup masa saja. This is the list of references that I use in writing my research proposal. And also this is my references And also this is my references. Before I end my presentation, a little bit of knowledge, which is my supervisor, which is Prof. Dr. Ho Wei Seng, that helped me during writing my research proposal. To my fellow friends that keep supporting me and help me in doing my proposal, my family that always give me a moral motivation and also supporting me doing this proposal, and also to my all lecturers that keep supporting me and always by my side when I need for help from them. I think that's all for me. Thank you.